Good day, everybody. We are still in the chapter 2. So after you study the cytoplasm and some membranes, so now we are moving to the endoplasmic reticulum. In O-level or SPM, you have learned the endoplasmic reticulums consist of rough endoplasmic reticulum and smooth endoplasmic reticulums. So today we'll continue to study what are the processes that found in the endoplasmic, the rough ER and the smooth ER, and the structure of this ER. Okay, before we start the ER, we must know what is endomembrane system. So endomembrane system is a dynamic relationship exists between the different parts of the cellular membrane system. That means it is talk about a membrane system inside cells. So the membrane molecules, that means the membrane molecules that are found in nuclear envelopes or the nucleus, they may appear in the rough ER, smooth ER, Golgi apparatus, lysosome, vesicle, and plasma membranes. So these interrelated membranes form the endomembrane system in a cell. So the function is to divide the cell into compartments. Again, it's into the compartment, and then allows different reactions occur simultaneously. Now, we look at this endomembrane system. So we start from the nucleus. So the nucleus here, because some parts of the membrane of the nucleus, they are connected with the rough endoplasmic reticulum. And then the membrane, the phospholipid molecule, or the, we call the phospholipid bilayers, they are not static, it is dynamic. Still remember what we have studied, the fluid mosaic model? So they are dynamic. So that means the membrane molecules, the phospholipid molecules, they may be found in the rough endoplasmic reticulum. Then they will be moved to the Golgi apparatus, and they may go to the lysosome. Where's the lysosome here? Okay, lysosome. Or they may be transported out and joined or fused with the plasma membrane. So therefore, this one, they form a membrane system we know as the endomembrane system. Endo is inside, internal. So the endomembrane system actually consists of the nucleus, endoplasmic reticulum, which including the rough ER and the smooth ER, Golgi apparatus, lysosome, vesicles, and also the plasma membrane. Okay, endoplasmic reticulum, as you know, there are two distinct types of ER. We call the rough ER and the smooth ER. Okay, so as you see here, this is the nuclear envelope, and then the outer membrane here, they are interconnected with the ER. So rough ER, they are ribosome attached to the membrane, whereas the smooth ER, there is no ribosomes attached to the membrane. Okay, rough ER consists of an interconnected system of membrane-bound flattened sac called cisternae. So compared to SCR here, look at the RER. This is known as the cisternae, and then the cisternae is a flattened sac. Okay, flattened. Okay, continue with the outer membrane of the nucleus, covered with many ribosomes on the outer surface of ER membrane. That's why we call it as a rough endoplasmic reticulum. So rough ER is concerned with modified proteins that are produced by ribosome to form glycoprotein, especially for extracellular enzymes by glycosylations. Then the proteins will be transported to the Golgi apparatus for further modifications. So this maybe is a new info for you. Okay, as you see this in this diagram, the This is ribosome. Okay, this is a cisternae, the lumen. Okay, rough ER lumen. So the polypeptides that forms are inserted into the lumen of the rough ER. Then glycosylations occurs. Okay, glycosylations occur in which they form a glycoproteins. Then after that, they will fall into the three-dimensional structure. Then they will bud off. Okay, bud off to form the transport vesicle. Then this transport vesicle will be transported to Golgi apparatus. Okay, so RER is abundant in cells which are rapidly growing or secretory cells, for example, the pancreatic cells, okay, and the meristematic cells as well. Okay, so this is the process of the glycosylations. The detail for the glycosylation, for this glycosylation is not required, but however, you must know the function of the RER. Okay, so you see the ribosome. This is an mRNA. So the polypeptides it form. Okay, they're inserted into the lumen of the rough ER. Then the glycosylations occur. So this is a carbohydrate, or we call it oligosaccharides. Okay, so they attach to the polypeptides, so to form the glycoproteins. Then later this polypeptide will be fall into the three-dimensional structure. They will be part of as in the transport vesicle. Okay, so smooth ER. When we compare to the rough ER, you can see the smooth ER is in the tubular form. Okay, not the flattened stack. It's a tubular form. So which is continuous with the nuclear envelope and the rough ER as well. Then another difference is lack of the smooth ER, lack of ribosomes, okay, compared to the rough ER. So SER's concerns with the lipid synthesis, non-protein substances, and some enzymes. So the lipids that form are phospholipid, cholesterol, and steroids. Then after that, these products will be transported to Golgi apparatus, okay, for secretions. So the examples are steroid hormones synthesized. That means there's in the cell they synthesize steroid hormones, for example, the reproductive organs and the adrenal cortex. So the SERs are active, okay, in producing steroid hormones. Okay. Or in liver cells, the SER, the smooth endoplasmic reticulums, also are very active because the enzyme that binds to this SER membrane, they're involved in the blood sugar regulations. So for your information, the hydrolysis of glycogen into glucose actually is carried out in the smooth ER. And also the detoxification of carcinogens, which is the cancer's causing agent. Okay, drugs and alcohol also are carried out in the smooth ER of the liver cells. In muscle cells, the smooth ER is modified into the sarcoplasmic reticulum. Okay, sarcoplasmic reticulums. So like this one, the purple color. Okay, cytoplasmic reticulum. So the function is to store and release calcium ions for muscle contractions. So you will study this the cytoplasmic reticulum, the role of the cytoplasmic reticulum in the next semester. So the overall function of endoplasmic reticulums are to divide the cytoplasm into more compartments for chemical reactions because they are separated by the membrane, and provide a large surface area for chemical reactions because they also consist of a lot of membrane. So this will provide a large surface area, okay, for the enzymes to attach to the membrane and to for chemical reactions. And also provide a pathway for transport of material through the cells, okay, from the rough ER to Golgi apparatus, or from smooth ER to Golgi apparatus. And for rough ER, The rough ER is to modify proteins that synthesized by ribosome and are transported to the Golgi apparatus, as you know, through the glycosylations. And for SCR is produced phospholipid and cholesterol. SCR also produces steroid-based compounds, that means the steroid hormones, for example, the testosterone, progesterone, estrogens, cortisol. And the SCR also breaks down the carcinogens and also detoxifies drugs and alcohol in the liver cells. Okay, so that's all for these videos that talk about the endoplasmic reticulums. So I hope you got anything, you can please contact me. Okay, so that's all. Bye.